How do archaeologists analyze microbotanical remains from ancient sites? Imagine tiny plant particles holding clues about how people lived thousands of years ago. That's what microbotanical remains are, small bits like pollen, phytoliths, and starch grains. Archaeologists use special methods to study these tiny clues and learn about ancient environments and diets. The process starts during excavation, where archaeologists carefully collect samples from sediments or artifacts like grinding stones. They handle these samples with care to avoid contamination, often sealing them in clean containers. Back in the lab, residues are extracted from artifacts using gentle brushing with sterile tools and distilled water. Sometimes, they also use dry methods to dislodge particles from surfaces. Once the residues are collected, scientists examine them under microscopes. Light microscopes help identify shapes and structures, while scanning electron microscopes give detailed surface images. Different microremains have unique features, like phytoliths shaped like tiny silica bodies or starch grains with specific patterns, that help identify the plant they came from. To understand where the remains came from, archaeologists map their distribution on tools and artifacts. This helps distinguish residues from natural deposits versus those from plant processing activities. They also compare the microremains to modern reference collections to identify plant species. Combining this with micro-wear analysis on tools, which looks at microscopic wear patterns, gives a clearer picture of how plants were used. To ensure accuracy, archaeologists include control samples from surrounding sediments to check for contamination. They store samples in sterile conditions and analyze them in specialized environmental archaeology labs. Microbotanical analysis can reveal what wild or domesticated plants people used for food, medicine, or materials. It also helps reconstruct ancient diets, seasonal activities, and plant processing techniques. This method complements larger plant remains that are visible to the naked, offering a more detailed view of plant use in the past. In short, archaeologists analyze microbotanical remains by carefully collecting samples, extracting residues, identifying them under microscopes, mapping their distribution, and combining findings with tool use studies. This approach helps piece together how ancient people interacted with their environment and what plants shaped their daily lives.